Imagine looking at a massive, highly complex construction site.
Okay, I'm picturing it.
Right, but the supply lines are completely severed. The site is basically starved of oxygen. The workers are exhausted, and everything is just, well, it's grinding to a halt.
Sounds like a total catastrophic failure.
Exactly. And normally to fix a failure like this, the instinct is to just, brute force a solution.
Right, bring in the heavy machinery.
Yeah, you send in massive fleets of trucks hauling raw materials or drop a giant crew of outside contractors into the middle of the site to rebuild it by force.
Which is incredibly risky.
It is, because the existing roads are crumbling. They might not even be able to handle the weight of those massive trucks. You risk causing a total collapse just trying to, deliver the help.
Right, you destroy the infrastructure you're trying to save.
Exactly. But what if you abandon the heavy machinery altogether? What if instead you just sent in millions of microscopic instruction manuals?
Oh, I like where this is going.
Right. Just tiny, almost weightless envelopes that slip right through the roadblocks, get handed directly to the local workers already on site, and give them the exact architectural blueprints to rebuild their own supply lines from the inside out.
What's fascinating here is that shift in strategy represents a fundamental restructuring of how modern science approaches cellular repair.
It really is a total pivot.
It is. We are moving away from forcing a damaged tissue to accept outside help, and instead, we are providing the highly specific genetic code it needs to orchestrate its own rescue.
Which brings us to today's topic. Welcome to the deep dive into the sources. If you are catching up on the frontiers of regenerative medicine, or if you're just relentlessly curious about how the human body can be reprogrammed, you are in the exact right place.
Absolutely.
Today's mission is to unpack a stack of dense medical and physiological research to really understand a revolutionary mechanism in modern biology. We are exploring how mesenchymal stem cell-derived exosomes,
Those microscopic instruction manuals.
exactly how they stimulate neo-angiogenesis, which is the growth of brand new blood vessels.
And specifically, we're looking at how they do this in ischemic or oxygen-starved pelvic tissues.
Right, by hacking a highly guarded cellular chain of command known as the AKTMTOR pathway.
It's just incredible science, because for decades, the entire field of regenerative medicine operated under a completely different paradigm.
The focus was always on the whole cells, right?
Yeah, exactly. The hyper-focus was entirely on the stem cells themselves. The overarching goal was to harvest whole, live, mesenchymal stem cells, or MSCs, culture them, and then transplant them directly into the damaged tissue.
The idea being that the cells were the medicine.
Right. The prevailing thought was that you drop the cells in, and they do the work.
Right.
But the research we are looking at today proves that the cells were essentially just, the manufacturing plant.
They were just the factory.
Precisely. The actual therapeutic value, like the true regenerative power, lies in the secretome.
The secretome. That's a great word.
It's the tiny extracellular vesicles that these stem cells manufacture and secrete out into their environment.
Which makes sense because using whole mesenchymal stem cells comes with, a staggering amount of logistical and biological baggage.
Absolutely. It's a nightmare logistically.
Right, because expanding autologous stem cells, taking a patient's own cells, and growing enough of them in a bioreactor for a therapy, that is an incredibly time-intensive process.
Very time-intensive and very expensive.
And you are dealing with fragile living entities that require complex biobanking, strict environmental controls.
Not to mention the biological risks.
Yeah, let's talk about that. Transplanting proliferating cells into a stressed environment carries inherent theoretical risks.
Like unwanted immune activation.
Exactly, or even worse, tumorigenicity, where the transplanted cells could, differentiate unpredictably and form ectopic tissue or tumors.
And that's why exosomes are such a breakthrough. They bypass nearly all of those hurdles.
Because they aren't whole cells.
Right. We are talking about nanoscale extracellular vesicles, measuring anywhere from 30 to 150 nanometers in diameter.
Which is unimaginably small.
It really is. And because they are cell-free, they do not divide. They cannot form tumors. They boast a highly favorable safety profile, optimal biocompatibility, and extremely low antigenicity.
Meaning the host's immune system basically just ignores them as they travel through the body.
Exactly. Furthermore, their infinitesimal size allows them to easily cross biological barriers, like the walls of capillaries, which whole cells simply cannot penetrate.
Okay, let's unpack this for a second because I want to push back on the physics of this.
Sure, go for it.
An exosome is essentially a tiny biological bubble, maxing out at, what, 150 nanometers?
Right. Very, very tiny.
And the extracellular space in a damaged, inflamed tissue is a highly chaotic, hostile environment. It's like a war zone.
It is a molecular war zone, yeah.
So how does a microscopic bubble survive that chaos without just dissolving or being instantly destroyed by enzymes before it even reaches its target?
Well, it survives because it isn't just a fragile soap bubble. Exosomes are enclosed in a very specific lipid bilayer.
Like a protective shell.
Exactly. And this lipid bilayer is remarkably similar to the lipid rafts found in the plasma membranes of our own cells.
Oh, interesting. So they blend in.
Yeah, and they aren't just sloughed off the surface of a stem cell haphazardly either. They're formed through a highly regulated inward budding process inside the parent cell.
Okay.
This creates these structures called multivesicular endosomes. When those endosomes fuse with the outer cell membrane, they release the exosomes.
So they're purpose-built for travel.
Precisely. That tough lipid bilayer protects the internal cargo from the hostile extracellular environment.
Yeah.
And because the exosome's membrane mimics the target cell's membrane, it can seamlessly fuse with a damaged cell.
It just docks right onto it.
It docks, fuses, and empties its contents directly into the cytoplasm, completely protecting the payload during transit.
It really is like a USB flash drive.
That's a perfect analogy.
Think about it. A flash drive has virtually no physical mass compared to a desktop computer, and it doesn't provide any hardware upgrades.
Right. It's not giving the computer a new hard drive.
Exactly. But the software code inside that drive can completely reprogram a massive supercomputer. The exosome doesn't need to provide the raw materials to rebuild the tissue.
Because it engages in horizontal transfer of genetic material.
Yes, it drops a densely packed, highly curated payload of messenger RNA, lipids, proteins, and crucial microRNAs directly into the target cell.
It instantly intercepts the cellular machinery.
And it forces a software update.
And that software update is most desperately needed in the ischemic pelvis.
Right, let's talk about the specific tissues here.
Ischemia is a severe restriction in blood supply, which leads to a catastrophic drop in oxygen and nutrients. When pelvic tissues lose their vascular networks, the architectural changes are devastating.
The sources highlight incredibly high-stakes clinical realities here. Like one prime example is Asherman syndrome.
A very severe condition.
Yeah. It involves trauma to the endometrium, which is the lining of the uterus. When the blood supply is compromised there, the healthy, highly vascularized lining basically dies off.
And it gets replaced by dense avascular scar tissue.
Right, intrauterine adhesions,
Yeah.
which physically obliterates the uterine cavity and is a leading cause of severe infertility.
Another focal point in the literature is cavernous nerve injury.
Which is a primary driver of erectile dysfunction following pelvic surgeries, right?
Exactly. In this scenario, the restriction of blood flow causes the smooth muscle cells of the corpus cavernosum to undergo apoptosis.
Programmed cell death.
Right. As the muscle dies, the body desperately tries to fill the void with collagen. So you lose the functional elastic vascular tissue and are left with rigid, non-functional fibrosis.
It just turns into scar tissue. And natural ovarian aging follows a similar ischemic decline, too, where restoring those microvascular networks is absolutely critical for preserving follicle counts and endocrine function.
The overarching theme across all of these is that the tissue is suffocating.
The oxygen supply is just gone.
And the only permanent solution is neo-angiogenesis. We have to build a new vascular network. But, and this is key, neo-angiogenesis isn't just laying down new pipes in an empty field.
It's not that simple.
Not at all. It is a highly aggressive, multi-step biological event. It actually requires controlled destruction before any construction can even begin.
Wait, destruction?
Yeah. The basement membrane surrounding the existing surviving blood vessels has to be enzymatically degraded.
So it has to tear down the old walls first?
Exactly. Matrix metalloproteinases step in and break down this tough outer barrier. Only when that membrane is breached can endothelial cells, the cells lining the inside of the blood vessels, break out of their current structure and migrate into the surrounding hypoxic tissue.
They migrate toward the areas of lowest oxygen, sprout new branches, and eventually two of these branches meet and fuse to form a functional capillary loop.
And once blood begins to flow through that new loop, oxygen is finally delivered.
And the vessel undergoes a maturation process to stabilize. We know this works too. The studies on cavernous nerve injury models show that injecting MSC-derived exosomes fundamentally reverses the pathology.
It isn't just minor symptom relief, it's actual regeneration.
Right. The exosomes increase the smooth muscle content. They elevate neuronal nitric oxide synthase, which drives vasodilation, and drastically improve the smooth muscle to collagen ratio.
They literally halt the scarring process and actively rebuild the lost vascular architecture.
We see the exact same regenerative architectural repair in endometrial models for Asherman's syndrome, too.
Yes, where the exosomes successfully orchestrate the formation of those beautifully branched capillary loops, completely restoring the thickness and function of the uterine lining.
So we know the exosomes arrive like little USB drives, and we know the end result is a newly minted capillary loop delivering life-saving oxygen.
Right.
But what exactly is the chemical switch the exosome flips inside the starving endothelial cell to initiate that aggressive migration and growth?
To understand the mechanism, we have to zoom way down to the intracellular signaling level and examine the AKT-mTOR pathway.
Okay, the alphabet soup. Walk us through it.
Well, when a tissue like the ischemic pelvis is starved of oxygen, the cells are under immense metabolic stress. And during periods of severe stress, a specific intracellular protein called PTN dominates the environment.
PTN.
Right. PTN acts as an aggressive molecular brake. Its primary function is to halt cellular proliferation and, in cases of severe hypoxia, actually promote apoptosis.
Wait, let's look at the logic of that for a second.
Sure.
If the tissue is actively starving for oxygen, and the only way to get oxygen is to build new blood vessels, why would the body deploy a protein to slam on the brakes and stop cell growth?
It seems completely backward, I know.
It does. If you are suffocating, the body should be desperately trying to build new vessels, not shutting the process down. It seems like a massive evolutionary flaw.
It definitely seems counterintuitive until you consider the alternative, which is cancer.
Oh.
Yeah. Hypoxia causes severe DNA damage and metabolic instability. PTN is actually a tumor suppressor gene. Its evolutionary priority is to prevent damaged, stressed cells from proliferating recklessly and forming tumors.
So it's a safety mechanism.
Exactly. From the body's perspective, it is much safer to force a starving, genetically unstable cell into apoptosis than to let it wildly replicate. PTN is basically playing it safe, choosing tissue death over malignant growth.
Okay, that makes perfect sense. The body is enforcing a strict quarantine.
Right.
So the endothelial cells are trapped. They have the potential to build new vessels, but PTN is keeping them on lockdown.
And this is where the exosome cargo completely changes the game.
Here's where it gets really interesting.
It really does. The exosome delivers a specific family of microRNAs directly into these stressed endothelial cells. Things like miR21-5P, miR144, and miR486-5P.
These are those curated software updates.
Yes. These microRNAs are highly targeted genetic sequences that bind directly to the cellular machinery and explicitly suppress the expression of PTN.
So they systematically dismantle the molecular brake.
Precisely. And once PTN is suppressed, the cellular quarantine is lifted, and a powerful signaling cascade known as the PI3KKT pathway is unleashed.
The PI3KKT pathway.
Got it. AKT is a central kinase that acts as the master regulator of cell survival. When AKT is activated, it immediately countermands the apoptosis orders, ensuring the endothelial cell survives the hypoxic environment.
It cancels the self-destruct sequence.
Exactly. But more importantly, AKT acts as the catalyst for the downstream growth pathways.
AKT tags in mTOR.
Right.
To clean up our construction analogy, if P10 is the bureaucratic red tape keeping the site shut down, the exosome's microRNA is the specialized permit that bypasses the bureaucracy.
I love that analogy.
And then AKT acts as the foreman blowing the whistle and waking the construction crew up.
Yes.
And MTOR, the mammalian target of rapamycin, is the heavy machinery. It is the excavator and the concrete mixer physically laying the foundation for the new structures.
That machinery operates by mediating the production of highly potent growth factors, most notably vascular endothelial growth factor, or VEGF.
VEGF is huge in this process.
It is. And MTOR also activates downstream transcription factors like STAT3. VEGF is secreted into the extracellular space, creating this chemical gradient that physically draws endothelial cells out of their existing vepals and guides their migration into the hypoxic zone.
And STAT3 and VEGF work in tandem to drive the rapid cellular proliferation required to actually construct the physical walls of the new endothelial tubes.
It is a brilliant elegant
It
cascades.
really is. But I mean, even the best heavy machinery in the world is useless if the ground you are trying to build on is toxic.
Right, the environment matters.
You can hit the AKTMTRA gas pedal all day long, but if the pelvic tissue environment is violently hostile, those delicate new capillary loops are never going to survive. So how do the exosomes prepare the tissue landscape to ensure this new vascular network can actually take root?
That is the million-dollar question, because the microenvironment is often the limiting factor in regenerative medicine. Ischemic tissues aren't just passive, oxygen-deprived zones.
They are highly active, inflamed battlegrounds.
Exactly. They're typically saturated with M1 macrophages.
And these immune cells are the body's first responders, but M1 macrophages are aggressively pro-inflammatory.
Yes, when M1 macrophages dominate a tissue, they release cytokines that stimulate local fibroblasts.
And those fibroblasts just go into overdrive, secreting massive amounts of extracellular matrix, predominantly collagen.
Which is the exact mechanism behind severe fibrosis. In Asherman syndrome, this is exactly how the intrarotorine adhesions form.
The M1 macrophages are essentially indiscriminate demolition crews. They are dumping toxic slag over the site, cementing over the entire area with thick, inflexible scar tissue.
Making any delicate architectural regeneration completely impossible.
Right. So what do the exosomes do about the M1s?
Well, the brilliance of MSC-derived exosomes is their pleiotropic nature. They don't just target one cell type.
Oh, they multitask.
Exactly. While they are delivering microRNAs to endothelial cells to trigger angiogenesis, they are simultaneously targeting the surrounding immune cells.
So they drop a different payload.
Yes, the exosomes deliver a different subset of genetic cargo, particularly sequences like the LET7B microRNA directly into the macrophages.
Okay, let's 7B.
And this specific microRNA intercepts a complex transcription factor pathway known as TLR4-NFB.
Okay, without getting bogged down in the alphabet soup, the crucial takeaway here is that this genetic interception forces the macrophage to undergo a complete phenotypic flip.
It forcefully converts the macrophage from the M1 pro-inflammatory state to the M2 anti-inflammatory state.
And the M2 macrophage is pro-regenerative.
Exactly. Instead of stimulating fibroblasts to dump collagen, M2 macrophages secrete factors that resolve inflammation and promote tissue remodeling.
They clean up the toxic debris.
They do. They stabilize the extracellular matrix.
So the exosomes are executing this coordinated dual-threat strategy. They operate as the peacemakers, delivering LET7B to flip the macrophages to the M2 state, halting the fibrosis and clearing the toxic slag from the construction site.
Right.
And simultaneously, they operate as the architects, delivering microRNA to the endothelial cells, suppressing the P10 brake, and engaging the AKT-mTOR machinery to sprout new blood vessels.
And neither process works in isolation.
They need each other.
Absolutely. The angiogenesis driven by MTR would completely fail without the anti-inflammatory environment cultivated by the M2 macrophage.
If we connect this to the bigger picture,
Oh, this dual-path paracrine signaling represents a total paradigm shift.
it really changes everything.
It does. We are no longer just treating the downstream symptoms of an ischemic pelvis. We aren't relying on surgical resection to cut away scar tissue in Asherman syndrome. And we aren't just relying on temporary chemical vasodilators for erectile dysfunction.
We are utilizing the cell's native nanovesicles to deliver a multi-pronged software update that commands the tissue to physiologically rebuild itself.
And the research indicates that we can actually manipulate the quality of that software update, which is just mind-blowing.
Wait, manipulate how?
Well, scientists have discovered that if you subject the parent stem cells to hypoxia preconditioning,
Meaning you grow the MSCs in a bioreactor with severely depleted oxygen levels.
right, like 0.5% or 2% oxygen, before you harvest their exosomes, it radically alters the therapeutic potency of the vesicles.
So by exposing the parent stem cells to hypoxia, or even inflammatory cytokines like TNF-alpha, you are activating their stress response pathways.
Specifically via HIF-1 alpha.
So you are artificially inducing panic in the stem cell factory.
That's a great way to put it.
By simulating the exact devastating conditions of the ischemic injury, you force the stem cells to change their manufacturing process.
Because it goes back to evolutionary survival mechanisms. When a stem cell senses an existential threat like severe oxygen deprivation, it stops packing standard daily maintenance kits.
It shifts all its resources into packing emergency trauma kits.
Exactly. The microRNA cargo packed into those preconditioned exosomes is explicitly supercharged to promote angiogenesis and modulate severe inflammation.
That is wild. So when you deliver those preconditioned exosomes to the ischemic tissue, The suppression of PTN and the subsequent activation of the AKTMT pathway is exponentially more potent.
The regenerative response is massively amplified because the genetic instructions were written specifically for a crisis environment.
We have covered incredible ground today. We moved past the logistical nightmares and theoretical risks of whole stem cell therapy to really understand the elegance of these 30-nanometer extracellular vesicles.
Just a beautiful system.
We explored the high stakes of ischemic pelvic diseases and how these exosomes bypass the cellular quarantine by suppressing PTN. We looked at how they hit the AKTMT or gas pedal to physically lay down new endothelial tubes, all while simultaneously reprogramming the immune microenvironment.
Forcing those macrophages to abandon their destructive inflammatory state and clear the path for regeneration.
The complexity of these mechanisms really highlights why regenerative medicine is moving so rapidly towards cell-free therapies. By speaking the native biological language of the tissue, we unlock healing pathways that simply cannot be replicated by traditional pharmacology.
It's the future of medicine, plain and simple.
Which leaves you with a fascinating final thought to mull over. We now know that inducing stress in the parent stem cell factory fundamentally changes the blueprints packed into the exosomes.
Right.
If we can manipulate the stem cells to pack specialized emergency trauma kits, how far are we from a future where this process is entirely bespoke? Imagine a scenario where a clinical team evaluates the precise nature of your specific ischemic injury, like the exact ratio of fibrosis to vascular collapse in your tissue. And then they custom engineer a specific microRNA sequence in a lab.
Tailoring the cargo.
Exactly. They could tailor the precise genetic code inside a batch of exosomes to perfectly match the unique vascular and immune deficits of your individual body, delivering the ultimate custom-written instruction manual directly to your cells. Thank you for joining us on this deep dive into the sources. Keep questioning the mechanisms, keep exploring the research, and we will see you next time as we continue to unpack the frontiers of what is possible.